we're going to continue our discussion about amino acids and proteins. Once you understand how peptides can be formed, the next thing is to understand that these peptides are not yet ready for them to perform their function. They should assume a specific structure in order for them to pursue their function. Structure of the amino acids determine the function of the protein. Proper folding is really essential for the function to be as required. There are different stages of folding. In this video, I'm going to talk about the primary and secondary structure, and in the next video, I'll talk about tertiary and quaternary structures. I put an asterisk next to quaternary because it's important to note that not all proteins have the quaternary structure, but almost all proteins go up to the tertiary structure. The primary structure in the protein folding is basically just the alignment of the amino acids in a linear chain. So this is where you'll determine the sequence of the protein. I just put in random amino acids, but just understand that this sequence really determines the overall folding, the overall function of the amino acids, and any mutations to this particular sequence could alter the function. The main bond holding this linear sequence of amino acids is a peptide bond, and that's almost all the interactions that you'll see in the primary structure. Once again, the primary structure is just a linear chain of amino acids, and you can use this to determine the sequence of the protein by various methods like DNA Sanger sequencing that we will talk about in future videos. When you read this, you would read this from the N-terminus because this is where you would have your amino group, and this is your C-terminus where you would have your carboxyl group. And if I were to add a new amino acid, I would add it on this side, or the C-terminus. Once the primary structure is formed, you'll get into your peptide will start arranging itself into the secondary structure, which is basically just local interactions between the amino acids. You are not worried about amino acids that are 3 or 4 positions down the chain, but these interactions are mostly with local amino acids that are right next to each other. You have 2 structures that determine a secondary, or 2 components that make up a secondary structure. They include the alpha helix and beta sheets. Alpha helix is a rod-like structure, and the peptide chains, they coil clockwise around it, around the central axis. I would encourage you to look up pictures for this. Beta sheets, on the other hand, are parallel or anti-parallel, and they're pleated. They're pleated in order to accumulate a high number of hydrogen bonds because the secondary structure is mostly characterized by intramolecular hydrogen bonding. There are no interactions between the R groups of the amino acids, and most of the interactions are going to be between the amino group and the carboxyl group. Both of them have a hydrogen bond between a carbonyl oxygen on one chain and the amide hydrogen atoms in the adjacent chain. Keratin, that is a fibrous structural protein that you'll find in your hair and nails, and that is usually made up of an alpha helix. Fibroin is made up of pleated sheets. Proline is a special amino acid because if introduced into alpha helices, it will create kinks and somehow not assist in the proper functioning of the enzyme. In alpha helices, it's important to note that the inside is called the helix core, and you will not find any R groups in that helix core. They will be pointing outward from the helix core. Proline is not usually found in alpha helices, but if it is, it's usually found in the end or at the beginning as a residue. Proline is flexible, so you would mostly find it in your turns of beta sheets. Beta sheets can be parallel or anti-parallel. If it's parallel, that would mean that the amino groups line up with each other for every single strand. If it's anti-parallel, it means that the amino group on one strand lines up with the carboxyl group on the other strand.